Hello, so I'm going to talk about the neuromuscular junction using this illustration. So a neuromuscular junction is the functional junction of a neuron and a muscle cell. So what I've done here is I've only drawn a part of the neuron. I've drawn the neuron's axon and the axon terminus. Here you have the muscle cell. There is the sarcomere you recognize. So let me identify some of the anatomical structures you see here, and then we'll talk about the process that's going to take place here. Because what's ultimately going to happen is this neuron is going to communicate to this muscle cell, telling this muscle to contract. And it's going to do that through the use of a neurotransmitter called acetylcholine. So here within the axon terminus, you see those purple round structures, and I've labeled them over on this side. Those are synaptic vesicles, because when we release a neurotransmitter onto an effector cell, that's considered a synapse. So we're going to see that term synaptic used in identifying some of these structures. So the synaptic vesicles found in the axon terminus contain the neurotransmitter acetylcholine, and so that is ACH for short. Here, and on this side, you see these little blue rectangles. Those are my illustration for a calcium ion channel. I've drawn an action potential moving down the axon. It's the portion of the neuron that generates the action potential is always the axon. Okay, when we move across to the muscle cell, you see that there's a space right here between the end of that neuron and where the sarcolemma begins here, and that's referred to as the synaptic cleft. So that's that small space in there. This is the sarcolemma of the muscle cell, and where it gets wavy right here, and you see these little guys, that region is referred to as the motor end plate. So what these little guys are at the motor end plate are acetylcholine or ACH receptors. So they're going to receive the acetylcholine, or in other words, the acetylcholine is going to bind to those receptors once ACH is present in the synaptic cleft. So we'll get to that in a minute. And then the sarcolemma comes along and dips down into the interior of the cell, and that is referred to as the T-tubule. Then these guys that you see that I've drawn on either side of the T-tubule are the sarcoplasmic reticulum, and so I've abbreviated here SR. The sarcoplasmic reticulum stores and releases calcium ions. So there's where I didn't label calcium ions. Those are those blue dots in there. And then there again you see the sarcomere, which consists of the thick and thin myofilaments. All right, so let's begin our process here. So when the brain decides it needs to contract a muscle, an action potential will be generated along the axon of a neuron. When the action potential reaches the terminus, it's going to open the calcium ion channels. Calcium ions are going to move into the terminus. So I'm going to draw some coming in here. So calcium ions are going to move in to the terminus. When they do that, the synaptic vesicles are going to move towards the neurolemma, at the end of the terminus. So let me find Okay. So these guys are going to move down to the end. They're going to fuse on to the neuron membrane there, and they're going to release the acetylcholine out into the cleft. That's referred to as exocytosis. Now, when the ACH enters the cleft, it will diffuse across the cleft and then bind to the acetylcholine receptors. When ACH binds to the receptor, that's going to trigger an event, which is going to generate an electrical event, or in other words, another action potential along the sarcolemma. So we're going to get another action potential here. I'm going to abbreviate AP. When that action potential hits the T-tubule, it's going to travel down the T-tubule. When it travels down the T-tubule, that's what's going to cause the sarcoplasmic reticulum to release its store of calcium. So that calcium is going to be released in response to that action potential moving down the T-tubule. That calcium is going to go to the sarcomere to trigger the sliding filament mechanism of muscle contraction. So if you remember that whole story where calcium binds to troponin, causing troponin to change its shape, removing tropomyosin off of the actin active sites, which allow the myosin heads to bind to the actin active site, and then they will power stroke. So that's another big concept. But there you have the neuromuscular junction.